Hello everyone, welcome to the lecture 7 on the series of excretory physiology. So in this class we are going to see on the concept of renal clearance. So the clearance mean or clearance refers to the rate at which a substance is removed, from the plasma. So as we all know, when the plasma is moving, if there is a substance X, the rate at which it is removed from the plasma is referred to be as clearance. So there are few terminologies to be remembered. There is a word called total clearance. Total clearance. Total clearance refers to a substance that is removed by all the organs of the body, or the removal of a substance by all the organs of the body is called total clearance. For example, you do have what is called metabolic clearance. Is a total clearance of a metabolite from all parts of the body. If a substance is removed from the plasma by a part of an organ or by any specific organ, then it is referred to be as specific clearance. Specific clearance by a particular. For example, here what we are talking about is the renal clearance or hepatic clearance. Hepatic clearance means it is a removal of the substance by the liver. Renal clearance, it is a removal of the substance from the plasma by the kidney. So what does exactly the renal clearance mean? It refers to the volume of the plasma that is completely cleared of a substance X by the kidney. Most importantly, per unit time. So if a substance that is there in the plasma, the amount of plasma that is moving to the kidney per unit time, in that per unit time, what will be the removal of this substance X by the kidney per unit time is referred to be as renal clearance. For example, if I say there is an increased renal clearance, it means there is complete removal of the substance from the plasma. If I say a substance has got a decreased renal clearance, it means there is incomplete removal of the substance from the plasma by the kidney per unit time. So this clearance can be calculated by the formula where clearance is equal to Ux into V by Px, where C stands for the clearance of the substance, Ux stands for the concentration of the substance X in the urine, whereas V dot stands for the volume of the urine that is formed per minute. Px refers to the concentration of the substance X in the plasma. So the clearance rate is expressed in ml per minute or liters per minute, where it is calculated by using this formula. So if the renal clearance is said to be as zero, if the renal clearance is said to be as 0, it means substance is not filtered at all. The substance is not removed by the kidney at all. That means there is no filtration. It is not at all filtered. Or for example, the substance is filtered. It might have been completely reabsorbed from the tubular fluid. So it may have been filtered in the glomerulus, but it would have been completely reabsorbed and it would have entered whenever the blood is leaving the kidney. So that is a possibility if the renal clearance is said to be as 0. If the renal clearance is more than 0, obviously the substances are filtered. It may be reabsorbed or it may have been secreted depending upon the need of the body. That is the reason why the renal clearance for that substance is more than 0. It is filtered, it is either reabsorbed or it may be secreted depending upon the need of the body. Here, most importantly, you should know about a substance called inulin. Inulin. Inulin is a polysaccharide. It is a fructose polymer which is less than 5000 daltons. Therefore, it is freely filtered. It is freely filtered, but it is neither reabsorbed nor secreted. So what does it mean? Whatever the amount is that is there in the filtrate is the same amount that could be seen in the urine. It is neither reabsorbed nor secreted. Therefore, whatever the amount that is filtered is equal to the amount that is seen in the urine. Therefore, it is considered to be as a best indicator of glomerular filtration. So, this is why inulin is referred to be as glomerular marker. Therefore, it is used glomerular marker. Therefore, it is used for the measurement of glomerular filtration. There is something called as clearance ratio. Clearance ratio. Clearance ratio refers to the clearance of the substance X to that of the clearance of inulin, the substance inulin. So if the clearance ratio is said to be 1, if it is 1, it states clearance of the substance X is similar or it is equal to the clearance of the substance inulin. If the clearance ratio is said to be less than 1, then it indicates clearance of the substance X is lesser than the clearance of the substance inulin. If the clearance ratio is more than 1, it states that clearance of the substance X is more than that of the inulin. So what does it mean? If the clearance is equal to the inulin, then that substance X can be used as glomerular marker. So it is neither reabsorbed nor secreted. So whatever that is the amount that is present in the plasma, it is seen in the urine. In the filtrate, it is seen in the urine. So what do you mean by less than 1? The substance is less than the inulin. That means to say the substance X is reabsorbed here. If it is more, what does it mean? The substance X is secreted. Then it is added in the urine. We have a substance called creatinine. Creatinine is produced from muscle creatine. It is a metabolic waste product. This is freely filtered. Little or it is not reabsorbed. Filtered freely. Little or not reabsorbed. So this can be used as an indicator for measurement of GFR or glomerular filtration. The advantage is that the creatinine is endogenously produced. It is produced inside the body, whereas inulin had to be injected inside the body, where creatinine is endogenously produced. Therefore, now there is a second substance called blood urea nitrogen, which is a waste product from the protein metabolism, is also a substance that is freely filtered, a little reabsorbed, and is also secreted. Therefore, the measurement of the plasma concentration of your creatinine and blood urea nitrogen helps us to know the functioning of the kidney. So if there is proper functioning of the kidney, if there is proper glomerular filtration, whatever the creatinine that is there will be filtered and it will be excreted out. If there is problem in the kidney, if there is problem in the glomerular filtration, there will be no proper filtration of the creatinine, which will be remaining as such. For example, the creatinine value in the plasma or in the blood is said to be as it ranges from 0.5 to 2 milligram per deciliter normally. If it is more than 3 or 4 milligram, or if it is more than 2 mg per deciliter, it means one of the reasons could be there is no proper filtration of the creatinine. So it is still present in the blood, which indicates the improper functioning of the kidney. There is another substance called para-aminohippuric acid. Para-aminohippuric acid. It is an organic acid which is freely filtered. It is not reabsorbed but secreted. It is secreted, not reabsorbed, freely filtered. Therefore, it has got more clearance ratio. Therefore, this substance can be used as an indicator for the measurement of renal plasma flow. Renal plasma flow is the best indicator for renal plasma flow. The point has to be remembered. So the next concept which we will be discussing is a concept called Tmax, or can also be represented as Tm, or transport maximum. Transport maximum. The transport maximum refers to, it is a maximum amount of a substance X that could be reabsorbed from the tubular fluid, or the tubular filtrate back to the plasma via the transporters. So we have seen, for example, if you take PCT, we have seen in the last classes. So in the PCT, we have seen the sodium is reabsorbed. Along with the sodium, glucose also goes along with the sodium through the co-transporter system, sodium glucose transporters, from the tubular filtrate to the epithelium, and it passes the basolateral membrane via glucose transporters. So the number of glucose transporters are fixed. So it can transport some amount of the glucose. If the amount of glucose increases than its maximum ability, it cannot bind and it cannot transport. So the channels or the transporters gets saturated. Every glucose will attach here. For example, hypothetically, for example, there are 20 glucose transporters. It is just an example for easy understanding. There are 20 glucose transporters. So if there is 10 glucose molecules that are available, it can be easily transported. So if there is 15, yes, it can easily. 20, yes, it is the maximum amount, 20 can be transported. But if it is more than 20, if it crosses its maximum limit, so here 20 can attach and it can be transported, the 5 remains as such. So this is referred to be as transport maximum. It is a maximum amount to which a substance can be reabsorbed through the transporters from the tubular fluid into the plasma. Taking example of glucose in specific. So the normal plasma glucose concentration is less than 200 milligram per deciliter, 200 mg per deciliter. If this is the condition, then all the filtered glucose will be reabsorbed. If, for example, the plasma glucose concentration crosses 200 milligram per deciliter, the glucose will be filtered, but all glucose that is filtered will not be reabsorbed. Why? Because there is saturation of glucose transporters. Therefore, this glucose will start appearing in the urine, or it will be excreted out. Or it will be start appearing in the filtrate. Mainly, first it starts appearing in the filtrate. For example, think if it is more than 300 mg per deciliter. Still more and more glucose will be in the filtrate, and it can also be seen in the urine. So it depends upon the GLUT transporters, the uniporters which is present in the basolateral membrane, whether it is saturated or not. Therefore, you do have, so this is referred to be as transport maximum. You do have another terminology called threshold level. Threshold level of a substance. It is defined as the plasma concentration at which the substance X is first excreted in the urine. First excreted in the urine. Therefore, you should remember a point, threshold level is always lower than that of the transport maximum. So the threshold level for the glucose here is 180 to 200 mg per deciliter. That means to say, if the plasma concentration is lesser than 180 mg per deciliter, no glucose will be seen in the urine. If it is in 200 milligram per deciliter, it will be first seen in the urine. If it is more than 300, definitely it will be seen in the urine. The point of the plasma concentration at which it will be first seen in the urine is called threshold level for glucose. So, threshold level refers to the concentration in the plasma, whereas your transport maximum refers to the substance that is present in the filtrate. So, in the filtrate, if the concentration is more, the transport is said to be saturated and it is minimal. So, this is the basic difference between the threshold level and the transport maximum. So, if the glucose is seen in the urine, then that condition is referred to be as glycosuria. or glucosuria, the presence of glucose in the urine, which is commonly seen in case of diabetes mellitus. So the next concept or the condition is referred to be as diabetes, which we are going to discuss is diabetes insipidus. So diabetes insipidus is a primary condition which is caused due to decreased levels of antidiuretic hormone. So then it is called as central diabetes insipidus, or there is a possibility that there is no or decreased number of V2 receptors. So we have seen in the explanation in the later videos, ADH act on the V2 receptors of the principal cells. They form aquaporins 2 and they help in the reabsorption of the water. So if there is no sufficient V2 receptors, ADH cannot act. Therefore it is called nephrogenic diabetes insipidus. Nephrogenic diabetes insipidus. So the diabetes insipidus is characterized by frequent urination, that is polyuria, polydipsia. Whenever there is polyuria, there is obviously polydipsia, increased water intake. Most importantly, it is hypoosmolar urine. So the urine is diluted. Normally the urine is hyperosmolar. It is 10 times more, it has got the osmolarity than the plasma. But in diabetes insipidus, it is more of watery, therefore it is diluted, therefore the osmolarity decreases, therefore it is hypoosmolar urine. The next point of discussion is something called diuretics. Diuretics. So diuresis mean loss of water in the urine. So more water in the urine is called diuresis. Antidiuresis mean decrease, no loss of water. I can put it as no loss of water or decreased amount of water loss. It is by water reabsorption in the nephron, mainly by ADH we have seen. Therefore it is called antidiuretic hormone. So what are diuretics? These are the chemical agents. These are the chemical agents which causes diuresis or the loss of water, or which prevents reabsorption of water. So where it can act? So wherever the regions where the water reabsorption is seen, there it can act upon. For example, it can act on PCT. So it can act on, most importantly, it will act on sodium potassium 2 chloride, which is blocked. So this is the one we have discussed in the last class, which maintains your, very important in the maintenance of counter current mechanism, and very important in the maintenance of medullary hyperosmolar interstitium, so that water can be reabsorbed. So if this is blocked, there is no counter current mechanism, or the medullary interstitium osmolarity is not maintained, there is no question of water reabsorption, then the water is seen in the urine. So then those agents are called loop diuretics. Loop diuretics because it is acting on the loop. Mainly this is seen in the ascending limb. So there is it can also act on the carbonic anhydrase. We have seen carbonic anhydrase, how it is one which helps in the hydration reaction. So if there is no carbonic anhydrase, if it is blocked, then the hydration reaction is blocked, and you can see increased amount of water in the urine. For example, you have osmotic diuresis, which is commonly seen in diabetes mellitus. So here in that we had seen in the transport maximum, if the amount of glucose is more, then the absorption is less. So whatever the amount of glucose that is present in the lumen, or in the filtrate will draw the fluid inside because of the osmosis. So it increases the osmolarity, therefore the water is drawn, so the increased amount of water is lost, therefore it is called osmotic diuresis. Or the diuretics can also act on the DCT, it can also act on collecting ducts. So many diuretics are being designed. So the next point which has to be known is a terminology called obligatory water loss. So in the first video we had seen about obligatory water reabsorption, which means the water is obliged to follow the sodium so that the water moves along with the sodium. So that is called obligatory water reabsorption. Obligatory water loss means it is the minimum amount of water that should be present in the urine for the removal of waste products that are produced. So it is the minimum amount of water that has to be there in the urine for the removal of the waste products that is normally formed inside the body.